So we just finished talking about the different ways that transcription can be controlled and that affects the different genes that are expressed. But it should be noted that there aren't that many control elements present in our eukaryotic cells. There's actually only about 12. However, that combination of control elements is responsible for activating transcription. So it's actually not the control elements themselves, not the enhancers or the repressors themselves, but the order they're placed in that affects whether a gene is going to be transcribed and therefore whether a protein is going to be produced. So only when those control elements are signaled and the appropriate activator proteins are present do we end up getting transcription of a gene. So, for example, this shows us an example of a liver cell and a lens cell, like a cell in your eye. And you can see both of them have their enhancers, and they both actually have this gray control element. The other ones are different, but they share this gray one. So why does a liver cell act like a liver cell and a lens cell act like a lens cell? It has to do with what activators are present. So in this case, when the appropriate activators are present for the liver cell, they're going to bind to that enhancer, the DNA is going to bend, we'll get our meteor protein and transcription factors and make it possible for RNA polymerase to bind and express the albumin gene, which is crucial to the liver cell. Notice, however, that the activators proteins are not available for the crystalline gene, which is expressed by the lens cell. So in this case, those activators are not going to be present in the liver cell. However, the crystalline gene is still present in the liver cell because it's the same DNA. But since those activators are not present, we're not going to get any transcription. Reverse it now. Notice the available activators are not going to bind to our enhancer for the albumin gene. That's the liver one. But they will bind to the enhancer for the crystalline gene, for the lens cell. Once again, our DNA bends over. We get our transcription factors, our mediator protein, and then our RNA polymerase is going to bind and transcribe the crystalline gene, but not the albumin gene. So that's why we can have cells with the same genome, the same DNA, express different things. And this is typically, these genes typically get expressed together. But what's very interesting about eukaryotic genes is they are not organized into operons as a group the way bacterial genes are. Genes that want to be co-expressed, like expressed at the same time in eukaryotes, could even be on different chromosomes, but they have the same combination of control elements. So the different activator proteins can recognize these specific control elements regardless of what chromosome they're on and actually promote simultaneous transcription of the genes. This transcription can be regulated by hormones or other chemical signals, and that's what kind of causes this all to happen. So that wraps up our ways that we can affect gene expression from transcription. And even though transcription is the most common place where gene expression is handled, there's also several mechanisms that happen after translation that can affect the expression of genes. And this typically happens as a result to environmental changes. So one thing that can happen is that our alternative RNA splicing can occur, and this is when we can get different mRNAs produced from the same pre-mRNA, just depending on what RNA segments we decide to cut out. So which ones we count as exons versus which are introns. And this is one of the things they've noticed, scientists have noticed, has allowed us to have such a large genome even though we don't necessarily produce a whole bunch of different types of mRNA. So for example, here's our original DNA, and then we have a transcription that occurred. So alternative splicing is an example of post-transcriptional gene expression. So after transcription, we get the different introns and exons, but notice with RNA splicing, from this same troponin gene, we could get an mRNA that looks like this, that includes the 3 gene, or we might get an mRNA that looks like this, the 4 gene. And depending on what was told to happen here, we can cut out the different parts with our RNA splicing and get 2 different mRNAs that are going to code for different genes. We can also get eukaryotic gene expression modification, differential gene expression, at the beginning of translation. So there are certain regulatory proteins that can bind to mRNA sequences or structures that will prevent the initiation of translation. And then after translation, proteins can be further processed and to influence gene expression. So certain things can happen. They could be cleaved, they could receive different chemical modifications, and also they might actually be marked for degradation. So if the protein it has been in the cell for too long, it can get a tag on it that's going to tell it now it's time to be destroyed. So those are just some of the ways that we can modify genes in the eukaryotic cell.